Hi there, I'm Dick Dixon, famous ghost hunter and reporter. And our cameraman slash woman today with us is...
Miss Cytosine.
Alright, and the people we're interviewing today are...
Hello, as you know me, I'm Miss Uracell, the crazy person. I'm back, and
I'm Miss Thymine, her weird, awkward friend.
Thymine Thunder, my best friend, man.
Alright.
Okay, let's get this interview started, Dick Dixon. I don't have much time.
So, can you explain to me what exactly protein synthesis means?
So, to put it simple, protein synthesis is pretty much means making proteins.
Alright. Okay, I like that. I don't like that.
Very simple for people who are not as educated.
It's not for me, of course, but for others.
Okay, Dick Dixon.
All right. Now, can you explain the base pair difference between DNA and RNA?
I would love to. Okay, so the differences between the base pairs, well, the first thing is DNA is a double strand. It has a double helix, right?
Double helix.
But RNA strand is only a single strand, as you see right here, one singular strand. Another difference is that RNA has a new person coming into
And it's your namesake.
It's my name, it's Uracell. That's how I got the name, because RNA for life. But so usually A, like ABBA, Gwen, however you call it, I don't really care about that one. But usually
my
it's
favorite band.
usually,
Mine is too.
Usually-
You have great taste.
usually it's A and T, so in DNA A and T paired together. But in RNA, A and U paired together.
A and U.
With Uracell, exactly. And then
So it's like Auska.
yeah, okay.
Simple way
And
to
then
put
the third difference, so as I mentioned, the first difference is about the, one of the differences is about the U one, the one is a single strand. And the third difference of RNA is that RNA has a ribose sugar.
Or ribose sugar.
And DNA does
Ribose.
not. DNA has some other
Ribose.
sugar, but I
Isn't that
can't.
called ribose?
Ribose sugar.
Yeah.
And DNA has another sugar, but I can't pronounce it. Starts with a D. Anyway. And you can easily
Diribose.
tell because DNA sugar starts with a D, RNA sugar starts with an R.
Fascinating. Now, this is a question right for, well, whoever wants to respond first. But what is the building block of protein?
You know building block, right, Donnie?
No, I definitely forget.
It starts with 2 A's, maybe.
Amino acids.
How did our cameraman know that?
Thank you, Miss Camera Woman.
All right.
Oh, it's okay, Donnie.
Really?
Thunder. I was trying to encourage her to
I
say
really
it.
appreciate it. I am very nervous on camera.
All right.
I was trying to encourage her. I knew it was amino acids, but I was trying to get my partner here to
Now,
say it.
can you talk to me about the 2 steps of
Yes.
protein synthesis?
So the two beautiful steps of protein synthesis are transcription and translation.
Ah, my aunt is a Bible translator. Real fact. So it's the same thing.
And is that it from now?
I think that might be it for this
Okay.
here portion.
Good.
Hi there. So can y'all explain to me just how this whole transcription process seems to take
Of
place?
course. So as you know, transcription is the first phase of protein synthesis. So transcription is the process when a molecule known as mRNA, also known as the messenger RNA, is formed by using a DNA template strand.
DNA template strand. Interesting.
Very, very interesting in my days. Anyway, so the first step of our beautiful transcription phase is initiation. So initiation is when a part of the DNA called the gene is going to slowly unzip.
I am a big fan of jeans. I wear them almost every day.
I know, it unzips like the zipper on your sweatshirt. Anyway, so once it unzips, that's gonna have a promoter sequence is going to allow, the beautiful promoter sequence is going to allow an enzyme called RNA polymerase II to attach itself to half of the parent strand, elephant. Okay, so now the second phase of transcription is elongation. So in the phase of elongation, the pre-mRNA is
Pretty sure it's
formed.
elongation, not elongation, but...
Elongation.
That's what you said.
That's what I said, Dick Dixon.
All right, well, maybe it's
Anyway,
your accent I can't
so-
understand.
Discrimination.
Just your-
Anyway,
Discriminated against.
in this phase, the second step of transcription, the pre-mRNA is formed, and the RNA polymerase II is going to assemble all of these beautiful RNA nucleotides complementary to the DNA strand. Yes, a beautiful thing right there. So you're gonna see how it's gonna slowly come together. And Dick Dixon, please start talking about something while I do this.
All right. When I was 6, I was abandoned on the side of the road and raised by a group of foxes. It was a very hard life, but it was worthwhile. I am not afraid, but people have often mistaken me for one. I've lived a sad life, but I believe I have found success in the modern world. There are some who say a man cannot be raised by foxes and both be successful and run a ghost hunting business. To them, I would say, look upon myself and I will show you greatness. This has been an inspirational quote with Dick Dixon.
Is the DNA done yet?
No. Dick Dixon, tell me your favorite color.
My favorite color is not turquoise. It might be purple. I'm currently deciding.
Okay, so we are just about done. This beautiful thing. Just a second. Ignore this, please. Okay, so we are just about done this phase of elongation. Elongation. So here, as you see, this is our beautiful mRNA strand. And as you can see, this now has a U for uracil because that's what it is for RNA. So this is the elongation phase. Now, the last phase of...
Don't laugh at her
Don't
while
just-
she's explaining things.
Wow. Anyway, you know, some people just can't pronounce things, okay? The reporter person, stupid person. Anyway. Is the camera facing towards me? Okay.
That was very mean. I
Okay,
must
thank
say
you very
so.
much. So, then the last phase of transcription is called termination. So what termination is the RNA polymerase is going to reach a terminator sequence, and that is where the terminator sequence of base pairs on the DNA strand. So right here, we're gonna see that this mRNA strand is gonna pull apart from the DNA strand, and it is going to go into the cytoplasm of the ribosome. But now, you got to keep in mind that before the mRNA leaves the nucleus, the caps on the ends are gonna be capped, and the intron, which is stuff that does not code, is going to be replaced. And the exon, which is stuff that does code, will stay the same. So now we're going to see that this beautiful mRNA strand is going to slowly move to the cytoplasm, which is where we're going to now start translation.
Fascinating stuff indeed. Just a few questions here. So, Miss Uracil, what is your favorite color? I feel bad because you asked me and I didn't ask you.
Oh, thank you very much, Dick Dixon. My favorite color has to be purple.
Purple. Good
Purple, and
choice.
sadly there's no purple base pairs right here, but it's okay.
A true tragedy.
Second favorite color is blue, and
Same.
again, blue isn't here right now because blue is tea. So that's also tragic.
So are you saying we discriminate against certain colors right on these parts?
I believe so, and in many other places we do as well.
Good Lord.
Okay,
This ain't
elephant.
right.
Okay, so the next beautiful step. Well, before we begin this, I would like to say that my best friend here, Thalmine Thunder, she was kind of messing around with the DNA, and maybe you saw there was a red one where it was supposed to be a phosphate. Well, that was her fault. I told her to assemble it.
My apologies.
She's my intern, okay? So
Yes, I'm so-
it's supposed to have a white one here, and then you probably saw two white in a row. There's supposed to actually be a red one here. So she just mixed the red and the white up. It's okay.
I'm sorry.
Anyway,
My fault.
it's okay, Fal. I mean, you're just learning.
Yeah.
Anyway, as Miss Uracil would like to start talking about translation. So translation is when a peptide is formed. So a peptide is what is adjacent between amino acids. So a peptide is formed, and it this wonderful peptide uses the genetic information of this mRNA, because the mRNA will tell you where the amino acids are supposed to go. So it uses the genetic information of the mRNA, and it starts at the AUG initiator sequence. Elephant. Okay, so then the following step, step 2 in translation, is when the 3 nucleotides of the mRNA, called a codon, are going to code for a specific amino acid. So then we're going to, here we have the transfer, the tRNA, the transfer RNA, and it is going to bind its anti-codon to the codon of the mRNA. So as you see, we are gonna codon of the mRNA, thank you camera person. So it's gonna go beautifully like that. And then now this one is just gonna fit in right here. Just gonna pop right in there, beautifully like that. Then we're gonna do it like that. And then as we see, this one now is going to pop off. Excuse me for my terribleness.
What are those black things?
This black thing, this is what the amino acid is.
What are
So-
those white things?
The white things is the phosphate. So then, what is it, cameraman?
Those
Cameraman,
are
what is
peptide
it?
bonds.
Yeah, that's what I meant. It's a peptide bond. It's adjacent to the amino acids. So then this tRNA is going to move out. This one's here now. And then this one is going to come in. It's going to fit into its anticodon. It's going to fit to the codon of the mRNA. It's going to go like this. And then this tRNA is going to move out again. And now we come to the third step of mRNA, termination. So once a beautiful codon stopper is going to reach the mRNA strand, it is going to stop this whole translation process. And then pretty much this, our beautiful amino acid, peptide bond, which is now a protein, is going to be released. Just like this. And there you have it, folks. That is beautiful protein synthesis.
Bye-bye.
Bye.
Excellent. Good work.
Excellent. We